actually have microscopes at your station. The tape or the slides will be taped into place. So you definitely don't want to be switching objectives or moving the slide around. So they're going to be in a particular area on a particular objective. And which objective I use will depend on the slide and what I'm trying to get you guys to identify. So this right here is the thyroid gland. This is at the lowest objective. And what you're looking at here, all of these circles, these are all of your follicles. And most of these circles have this pink material in them, or sometimes it looks orange, and that's the colloidal material. If you have a follicle that doesn't have that material in there, then it just means that that follicle has probably recently released thyroid hormones. And so it needs to build that colloidal material back up. But in between the follicles, this is where you would find the parafollicular cells or the C cells. So for this one, it's going to be better to magnify it because then you can actually see cells. Because here you can see the outline of the cells, but you can't really see the cells. So I'm going to take it up to 10x. So this is at 40x. And so what you're looking at, this would be a follicle right here. This would be a follicle. This would be a follicle. This would be a follicle. And this would be a follicle. So the follicles are always going to be lined with simple cuboidal epithelia. So if you find this, follow that, there would be simple cuboidal epithelia around all of them. And then the cells that are in between the follicles would be your C cells. So again, if this is a follicle, it's going to be lined with simple cuboidal epithelia that would be going around like this. And then if this is another follicle here, it's lined with simple cuboidal epithelia. This one lined with simple cuboidal epithelia. And then anywhere in between, so these cells here are in between this follicle and that follicle. These would be your C cells or your parafollicular cells. So these guys would be releasing calcitonin. These would be releasing your thyroid hormone. And so for that one, it's going to be best to see it at either 10x or 40x. We were just looking at 40x. This is 10x. You can also see it nicely here too. So again, this would be a follicle. This is simple cuboidal epithelia going around. This would be another follicle, simple cuboidal epithelia, going around like that. And then these cells in here that are in between that follicle, and that follicle, and that follicle, those would be C cells, or parafollicular cells. Okay, so that's the thyroid gland. Go over to the pancreas. So for the pancreas, let's get it in a good spot here. So this is at 4x, and when you look at the pancreas, most of what you're looking at, all of these purple cells out here, these ones that are staining really dark, those are your acinar or your acini cells. And those are your exocrine cells. They're the ones that are making the digestive enzymes. These lines, these kind of white areas that you're seeing, these are capillaries or small blood vessels that are cut lengthwise. the smaller white lines that you see in here, or the white spaces, those would be smaller vessels or more likely capillaries in here. What you want to look for are the islets. The islets are the endocrine cells. And there aren't going to be a lot of islets, but what you're looking for are a group of cells amidst all of these acinar cells that are clumped together and stained lighter. So they usually stain a lighter pink color. And usually the islets are going to have a lot of capillaries in them or right around them, because that's where the hormones are going to go, are into those blood vessels. So this group of cells right here, these are islets. This is an islet. So in that group of cells right here would be alpha and beta cells, cells that are making, alpha cells are making glucagon, beta cells are making insulin. And you can see this islet here has a lot of like white, looks like white space in it. And those are just capillaries that are running through and around the islet. Then you'll also see ducts. So these round guys, generally lined with simple cuboidal epithelia, those would just be your pancreatic ducts. So all of the digestive enzymes would get exocytosed into these ducts, and they would all converge to form one big pancreatic duct that then went over to the small intestine. So acinar cells or acini cells, these lighter areas are your islets. Inside of them are alpha and beta cells. You can't tell the difference between the two. It's too hard to tell. But just know that alpha and beta cells are making glucagon and insulin. If we magnify this, so again, this is at 4x. If we would go up to 10x, this is a good magnification too. You should still be able to see this is your islets. It's a group of cells that are staining a little bit lighter than these other cells out here. And you can see that there are capillaries running around it and then within it. So this would be the islet that we were just looking at. Out here would be your acinar or your acini cells. And those would probably, going up higher in magnification, then you're going to start to have a hard time seeing the difference between islets and the acinar cells. So 4x, 10x would be the best to see that difference. Okay. So then if we go over to the adrenal gland, and I generally think the adrenal gland is one of the harder slides. And I think it's harder just because it's challenging to see the different cell types, particularly magnifying them like this. You lose a lot of the definition that you can see with your microscope. But when you look at the adrenal gland, so this would be at 4x, again, what I try to do is just get a sense of where's the capsule, where's the medulla. So out here, all of this stuff out here is the capsule. It's all part of the capsule. And so is these, kind of looks like strings running in this direction. That's all part of the capsule, which is just dense connective tissue surrounding the whole organ. And then you can kind of see there's a layer of cells that are running about the width of my hand right here. And that's the layer of cells that's right below the capsule, and that's your zona glomerulosa. And again, you kind of lose the definition here projecting it, but when you look, to me, those cells kind of look like they're kind of swirling a little bit like this. Kind of like that. Here you can definitely see them kind of moving in that direction. But the layer that's right under the capsule, that's zona glomerulosa, that's the layer that's releasing your mineralocorticoids like aldosterone. And then you go into the next layer that's going from about here down to about here. That's your zona fasciculata. And these cells are kind of running like this. it's definitely gonna be your biggest layer. And again, I use the capsule and the medulla as a point of reference. The cells right below the capsule are glomerulosa. The cells right above or on either side of the medulla are reticularis, which we can't see here. And then this big group of cells would be fasciculata. So if I showed you that, and if I put the pointer right here, then that would be glomerulosa. If I put the pointer out here, that would be the capsule. If I put the pointer kind of right in the middle here, that would be fasciculata. So now I'm going to go down, and fasciculata releases the glucocorticoids like cortisol. And I'm going to keep going down. And here you can see the medulla. So this area that looks more purple, this is all the medulla right here. That's all the medulla. And again, the layer of cells on either side of the medulla, so that would be these cells right here, this is reticularis. And when you look at these cells, again, it's not a real big, thick layer. Glomerulosa is going to be the smallest layer, then reticularis is a little bit bigger than that, fasciculata is the biggest layer. But when you look at this layer, you should definitely see a color switch between these that are pinker, these that are more purple. And the other thing about reticularis is the cells are kind of going out at angles a little bit instead of running straight up and down or being kind of swirly. They kind of radiate out a little bit. And reticularis is going to have more spaces in here, lots of capillaries in that particular layer. So if we keep moving down, so again, this would all be medulla right here. And then again, you should see a color switch between here and here. So if that's medulla, then you go reticularis again. So this layer of cells right here, this is reticularis. And then down here is going to be fasciculata. And then if we keep moving down, okay. So medulla is right here. Again, you can see the boundary. That's the edge of the medulla. Then the layer of cells right beside the medulla would be reticularis. Down here's the capsule again, and that layer of cells right by the capsule is glomerulosa. Here. And then all of these cells here in the middle, that's fasciculata.
What's the area that's kind of jutting inward like that? That weird pink thing?
This right here
Yeah.
is probably just the tissue probably folded when they were staining it, so it looks a little bit darker. But you should be able to kind of see that glomerulosa looks a little bit darker, and that's because the cells are kind of clumped together a little bit because they're kind of oriented like this. So they're kind of close together. Fasciculata, the cells are more like this. And then reticularis, they kind of go out a little bit like this, and there's more space in it. So I'll either show you this at 4x or 10x. If I were to show you at 4x, I would definitely make sure that you could either see the capsule or you could see the medulla so that you can always reference it. You see the capsule, cells right beside it are glomerulosa. You see the medulla, cells right beside it are reticularis. Anything in the middle, fasciculata. Questions on endocrine?
Can you go up to 10x on that?
Yep. So 10x, let me go to the top here.
It seems like it would be harder to see the layers.
It's going to be hard projecting it like this. So this is at the top again, so this